Well, thank you for joining us. I'm going to talk today about the NASA System Biology Platform for Space Omics Repository, Analysis and Visualization called GeneLab. My name is Sylvain Coste. I am the project manager for GeneLab. The talk will be divided into two parts. I will be having Amanda Saravia-Butler, who is a GeneLab data processing lead, describe to you how to use our analytical tool. So I will spend about 30 minutes on the system itself, the online system, and giving you some ideas of why GeneLab was created in the first place. Then Amanda will walk you through the analysis platform that we put together. Because it is a recording, we had some technical issue with the live demo of this webinar. There will not be any question and answer here. I apologize for this. You can also find out at the bottom of this slide here our Twitter handle and our website. So why GeneLab? Why did NASA spend time and energy to build this database? So every 10 years, the Academy of Sciences create what we call the National Research Council, which is a panel of experts that give advice and suggestion about where NASA should be putting some effort. And the last decadal survey, which was in 2011, really identified all the omics as a new technology that's going to really change and revolutionize our understanding of the effect of space flight on life, on biological system. And back then in 2011, on this diagram here, this is the classic, you've seen this curve probably many times, it's taken from NIH. It's the cost of doing DNA sequencing as a function of years. And you can see that for many years, that cost was following the Moore's law, basically suggesting that it was highly driven by the computer cost. But in 2007, the technology of sequencing changed drastically. And when that happened, the cost went off that curve. And in 2011, the cost was extremely low already for sequencing. Now, we're in 2020, and basically, if you look at where we are now, the cost has dropped for another log 10. So it's 10 times cheaper than it was 10 years ago. And 10 years ago, the omics and the sequencing was already considered by our experts being a key feature of space exploration in many ways, more in ways of understanding how life changes in space, but also how we can prepare better to live in space. And that's what the GeneLab database is about, providing this information. So if I were to, this is a bit of a cartoon description of GeneLab. On the right, you see this cloud. This is where our data stands, and they are sitting there like many different databases that are doing omics. And I'm listing here MGRAS, EBI, and GEO from NIH. And so just to name one, GEO, for instance, it's the database from NIH to have gene expression data. And the reason we put those three there is because we have been federating with them. I will show you in a minute what it means. But GeneLab is just another of these databases, very specialized to anything that is spaceflight relevant. And I will try to explain to you in a minute what it means. Something quite unique to GeneLab, the team that we have, compared to any other database that you would know, is that we also benefit from a sample processing lab that actually can take experiment conducted on the space station and or other transport system. And once a principal investigator is doing an experiment in space, we can then they don't necessarily use all the tissue that they had, and so we can then get these tissues and process them for additional analysis, such as RNA sequencing or proteomics or epigenomics. And so the GeneLab team is actually doing this on a yearly basis. We even have now a center a facility that can do sequencing on the site. So we have a very good control of what's happening. So that's quite unique to us. The other source of data is clearly NASA PI or ESA PI that wants to upload their their data into GeneLab. We have a web portal and a workspace. I won't talk much about the workspace today. And then we have an analysis toolshed that Amanda is going to describe. What we have too is what we call the analysis working groups. Those are individuals, scientists that have joined us. I will show you the link in a minute. And together we've been really trying to turn the raw data of omics into something more meaningful. And so initially, GeneLab was just an archive system for raw data. It has moved since then towards also providing processed data. Raw omics data tend to be extremely big. It takes a long time to extract the information, and the process is always the same. So with the analysis working groups, all these scientist experts that have been working with us, we decided on platform and pipeline that really turned this raw data into something more meaningful. And this is what we also share on our website. This helps you get new knowledge from space flight information. And the goal here is to really democratize the access of this information from the data scientist who knows how to analyze this data all the way to a citizen scientist who is going to spend more time on those processed data and just trying to figure out what's happening. To facilitate the visualization of the processed data, we have created what we call the visualization portal. And I will walk you through this as well. This is a classic slide from NASA. It's called the NASA Space Tracers. On the right here, you can see there's 5 of them. The distance from the Earth is a tracer, and you can imagine that as we move to the Moon and to Mars, which are our big goals, the Moon is actually happening in a few years, and Mars exploration is also happening maybe in 10, 20 years. So those are our targets. And obviously, distance is going to be a big stressors for these missions, as it takes longer and longer to communicate with people on these different on Mars, for instance, or on the Moon. Confinement is another big issue. People are going to be confined for a long time, away from everyone. The environment under which they operate is not. It's a bit of a hostile environment. There are challenges in terms of the air that they breathe. The carbon dioxide is much higher than what we have on Earth. So there are challenges. I'm going to highlight the 2 challenges that have been studied extremely well on the ground as well. It's radiation and the gravity. So why do I bring this up? On the left here, I can see a pie chart of the type of study we have in GeneLab, and you will see that the majority are space flight, but we also have experiments related to ground experiments. So experiments that were never flown. And typically those experiments are made primarily of simulated cosmic radiation. This is done typically by going to an accelerator. So all the American principal investigators typically do these experiments at Brookhaven National Lab in Long Island, where we have a large accelerator, and there is the NASA Space Research Lab. And then we also simulate microgravity in different ways. I will walk you through in a minute. So if we focus on those ground studies and we see the pie chart of what they are made of, 45% of those ground studies are really cosmic radiation simulation. But then we have about 26% that are simulated microgravity. And simulated microgravity are typically either cell culture that are grown into in suspension, and then we actually have a perpetual motions using something called clinostat. Or if we use animals instead of cell culture, then the animals are typically uplifted, and we make sure that their hind limb is not getting any load on the ground. And so that's called the hind limb unloading experiment. And those are a way to simulate the loss of load when you would be in space. The human analog to this would be a bed rest study, when we have people resting into a bed and we see how the muscle dystrophy is taking place and what kind of biological changes we see in using different omics tools. So those are the type of ground study we have. So the reason I bring this up here is that if you are interested in uploading some of your data, think about those 5 stressors, and if your experimental design addresses one of those stressors, feel free to reach out to us and to upload your data. Okay. Basically, the philosophy in terms of the animals that we have is really, we fly all kinds of animals on the space station right now, and from large being a mouse, this is the largest we have. Really, there's a few rat experiments in the past, but it's very difficult to fly large animals on the space station, and it's much easier to fly small animals like C. elegans and Drosophila. Of course, when you use small animals, you can have more animals being flown at the same time. And so we can really address we can look at the importance of genes for a specific response by having strains that have certain genes that are deficient. With mice, it's much harder to do this experiment because we can typically fly 20 to 40 animals at a time. So mice are typically only one strain of mouse, and then we really are looking more at a physiological response for these animals. The beauty of the mice is that they're very close to us, so they're more relevant to human. The goal here is to really use this system to infer information for the human mission, and then typically the Drosophila, for instance, to take one of those small animals, have been shown to be very helpful to look at the immunity, the immune response, the circadian rhythm, neural behavior. So they are actually quite a nifty system, even though they are, very different from us. Genetically, they still bring a lot of information that are very relevant to human. We don't only have animals in the database, we also have microbes and we also have plants. For the microbe, it's typically either swab that were done in the space station to see what grows over time in the space station, or we also have microbes that are literally incubated in the space station to see how they grow differently in space, so with very controlled conditions. Plants, I'll walk you through one data set where we look at basically how plants can grow in space without gravity, so in microgravity, and also looking at all kinds of things like the light cycles. All these are defining an ecosystem in space that will be much more constrained than the ecosystem we have on Earth. And the importance here is to really start thinking about how those different groups talk to each other. The same information now in a graph manner. In terms of organism, like I was saying, a big chunk of our data are rodents, and mainly mice and some rats. But we also have 20% of our data being human, either human studies that are not astronaut relevant, like a bed rest study, or in terms of other human data, it will be human cells grown in space. We have plants, microbes, fruit flies, fish, invertebrates. A total of 244 studies as of today, May 6, 2020. In terms of the type of assay we're looking at, we primarily focus on transcriptomics, so gene expression with microarray and RNA sequencing. But we are really covering, like I said earlier, all the omics that we know. So we have epigenomics, like methylation profiling, metagenomics, proteomics, RNA methylation is even in there. So we're really covering the full spectrum of omics at the moment. Something very important that you may have heard in the past is the FAIR principle for database. So FAIR stands for findable, accessible, interoperable, and reusable. And I'm glad to say that, in 2018, we published this manuscript comparing our fairness with respect to other big databases out there like Geo, ENA, MGRAST. And, out of those 4 principles, we have the highest score. We have an advantage over the older databases because we are a new database. We can really implement immediately at the building of at the development stage of GeneLab. We were able to embrace many of the concept of FAIR. So findable means that you need to be able to facilitate the search of the resources in the system. And I'll walk you through a couple of those findable aspects. Accessibility is really the ability to retrieve using an ID, standard protocol, metadata. The interoperable aspect of the database is really to use formal, broadly applicable languages to really start to see how we can exchange between different databases. And reusable, again, is very much focused on the description of your metadata. The better we harmonize the metadata and the data into the database, and the easier it is for someone to reuse this information. So it's really important. So without further ado, those are the different studies I want to walk you through in GeneLab. So I'm going to escape this and I'm going to go to GeneLab. So genelab.nasa.gov, that's the website. If you arrive to GeneLab, you will see basically different things. So those are the news. You can see that's today's talk. That was the advertisement. We've also been involved with our analysis working group. I've been involved with a group called COVERED, which is COVID-19 International Research Team. And there was a nice workshop a few weeks ago with, basically some members of our analysis working group basically doing some COVID work. Talking about the working group, you can find information here under AWG. We have the charter, we have the different groups, and some information about the workshop we have. And you can join us by clicking here. Now, those are the main buttons I want to go over. This is the link to the data repository. This is the link to the workspace. That's if you want to submit data. The workspace and the submission portals are related to each other. We are about to release a new submission portal that would really load your submission through the workspace. So the workspace is used to load omics data and to share with other investigators. So you can create an account today and you will be given 30 gigabytes of space to work with. And it's also the platform we use to let people share their information with others and also to submit to our database through a careful curation. The analyze data is the button that takes you to the analytical toolshed, and Amanda will walk you through this. This is an important link. It's the environmental page. It describes how we collect all the telemetry for the experiment. So if you're in the space station, we will give you the radiation dosimetry, so how much dose the sample received in space, with some description about the different type of cosmic radiation that deliver the dose. We also have information about carbon dioxide level of the specimen. So if you have a mouse, you will see that maybe they get 600 ppm to 3000 ppm compared to on Earth, we're around 350. Temperature, humidity, light cycles, all this information is described in the environmental data. And finally, the visualization of the data is linked to the data repository. You can either visualize directly by clicking here, but I will walk you through it through the data repository. So let's go to the data repository. When you arrive there, by default, GeneLab is selected. And then if GeneLab is selected, we are benefiting from this rich metadata ontology search capability that you would not have if you were into another website. So this is part of our fair findable, the F factor. So for instance, I can go there and I say, I only want to see your space flight sample. So right now you see there's 244 studies. If I select space flight, and now you drop to 140. Now we can go further down and say, I want to only look at a mouse. Well, let's just do mouse first. So now we drop to 52 studies. What's interesting is you can see here the actual search, and we can use Boolean. So for instance, let's switch to ground study instead. And I want to look only at the radiation. So there's 27 mouse study with ground studies. And I can search only the radiation analogs. So I've got 45 experiments that have radiation, that are ground study, and then now I can actually put and mouse. It's important to put the Boolean term you're using for searching, and or you need to use it in uppercase for it to work. Now I'm down to 16 studies. Okay. If I click NIH-GEO, I will lose all these factors. Another thing I forgot to mention is, so let me show you. If I go there now, it's just going to search radiation and mouse, both in GEO and GeneLab. You know you're in GEO when you see this icon. So those are experiments that were actually from GEO, and some of those are GeneLab experiment. So there's 150 experiments that were found with the keyword radiation and mouse in GEO and GeneLab. Data are duplicated in GeneLab and GEO for anything spaceflight relevant that existed in the past. In GEO, we have ingested this information into our database. Let me remove the NIH-GEO for now, and let me show you a few more features that are interesting. So if I go back here and I look for let's say spaceflight, I can actually now look for a specific organism. I'm going to take Arabidopsis, which is a plant. And I see the 17 experiments that were flown in space that use Arabidopsis. If I navigate through them, I'm going to show you one that's very interesting, is the this one. So in that study, they looked at a lot of different cofactors. So what we call factors in our database is really things that are changed in your experimental design. So in this case, again, by the way, this is part of the F statement, fair. Basically, you have rich metadata where we are controlling the fact that here they are, in that study design, they were ecotype. And you can see here what the definition of ecotype is: a biotype resulting from selection of in a particular habitat. So the habitat was tested in this experiment. Space flight was another factor, and the light treatment. So basically, they were looking at the impact of light. And if you look at this experiment, what's interesting is we have this tab system here where you can navigate through. So you have the description, and typically at the end of the description, you can actually download the data set through a data citation, either as a BIP file or as a RIS file. So that can then be used for basically, this is a classic things that you want to upload into your EndNote reference system if you have EndNote or Mendelian or something like this. And then what we've been recommending people to do is to, when they write a manuscript using some of the data from GeneLab, they should cite the data itself, and you can do that with that system. You have a description of the protocol that really walks you through all kinds of things, from the library construction to some, how the plants were grown. Quite an elaborate amount of information here. The sample level, you can actually basically control your columns and the width. And so here we have those two columns are always there, the source name and the sample name. So source name tend to be more elaborate, and sample name is typically shorter. That just gives you the name of the file. You can make the column wider if you want. And then you can navigate through, toggle through this information. Duration of this harvest time here, 11 days, still the same. So that's why it's not a factor. The type of preservation, RNA later. And then if you want to select some of this information, you can just say select export column, and you can select or unselect all and just decide what you want to export through CSV. Okay. Assay looks very similar, except that it's really just focusing on the assay with their name. There's quite a few files here, as you can see. And on this tab here, you can select the type of assay you want. So here, there's only one assay, transcriptomic. So that's the reason. Finally, there is a publication link that shows you what that data set, where it was published. You don't always have a publication associated to the data. For instance, the data we produce at the beginning there's no citation because they are freely available for anyone to analyze. Finally, something very interesting is the study file tab, and then in there you can basically navigate through the files you want. So for instance, you can see that data set has been already processed. You can basically navigate through the different data. So you can look at the RNA-seq, for instance, or the process file. And the process file goes all the way from, normalized count. I should say it goes all the way from, trim data to normalized count and differential expression analysis data. So quite a few. You can also see these. This is probably the better way to download files. You see, you can select all and here for selection and downloading. You can always see these files in the assay section by navigating through them. There's quite a few here, and you can actually see their name at the if you move to the right here, all the different files. So you can see it's a different way of looking at the file, but you have the QC files, multi-QC, single QCs for different steps, from trimming to counts and so on. So and then again, Amanda is going to walk you through the pipeline to generate all these process files. Now, final one is the visualization. You can click there and it will take you to the visualization of that data set. I believe I already looked at it. So let me see. Here we go. So the data set is pretty complex. I'm not going to walk you through it. They looked at different impact of light. This is a PCA plot. That's where you land typically on a PCA plot. I think for simplicity, let's walk over a data set that is a bit easier to understand. Okay, so we are back to the database. Here I remove all my I clear all my filter, and now let's search for a flight mission using mice. So I'm going to put space flight, and actually you can select the mouse here, but I'm just going to type mouse instead. So there's 52 of them, and let's say I want an muscle. The reason I picked muscle is because there's, microgravity will really have an impact on the muscle, and so it's obvious tissue to look at. And we're going to pick this one. And the soleus muscle actually is that study is very nice. It's the Rodent Research 1 mission. If you click on that, again, we worked. Description is there. Rodent Research is actually a NASA Ames project that's been going on for many years. The project scientist is Ruth Globus, so that her name is shown up here as the PI for this work. There's a description of what is Rodent Research. It's basically a mass experiment being flown on a specific habitat on the space station. This data set is really interesting. It has both epigenomic using DNA methylation profiling, and it also has transcriptomic data with RNA-seq. And let's see, those are the protocols. So again, description of the mass habitat, how the tissue was collected, library preparation, and so on. In terms of the name, something to consider here. I'm sorry, going back to description. The only factor is spaceflight, by the way. So it's a simpler experiment in terms of design compared to the plant we were just looking at. If we click on sample again, you can actually see from the name itself, GC stands for ground control. And what you can see is that there's 6 ground control, and 6 flight. And that information, the space flight is that factor is shown here as space flight. And you can see those samples are space flight, and those are ground control. Okay, and you have the duration, 37 days in space. Euthazel was used for euthanasia, and all kind of information here. Again, if we go publication-wise, there was one study done on this data set by our team, GeneLab. But again, this data was really one of these data augmentation I was mentioning. So they were put online for quite a while, and it was up to people to look at the data and analyze them. And right now, the study that was done was really focusing on microRNA signatures, but it's open for people to look at all kind of things on this data. So feel free to go there. All the process data are available here. I walk through this data with you on the previous data set. So with the plant, so I'm not going to dive there, but let's focus on visualization. So when you visualize data, they will look like this. It's loading. So when you arrive on the visualization, this is what happened. You get that PCA plot. That's a default landing page. And the PCA plot is really a way to see how the different group considered here are, their gene expression signatures are similar. So it's really a way to see how clustered the data are. And then so if you see this cluster here, what you can clearly see is that the green points are all the six animals that were analyzed from the ground control, and you can see how well clustered they are. So that means that their gene expression are very similar. In contrast, the animals that were flown in space have a bigger variation. But one thing for sure, if you look at PC1 and PC2, there's a very strong separation in terms of their components, and even PC3. So very quickly, I can already see here that if I were to compare those points and those points, I would see a much stronger reproducibility in this data. And also I can see that there's going to be large differences in terms of gene expression. That can be visualized quickly with the query data. So if you go there and you remove all your filter, and we set the p-value to 0.05, and I ask for only 10 genes maximum, you can see that you basically have many genes that are highly significant. That's the p-value for those genes and the fold change. And you can see that you have a large amount of genes that are significantly different between flight and ground control. You control the comparison here. In this case, because there's only two conditions, flight and ground control, you don't have much of a choice. You can also visualize this data by typing your favorite gene. So let's look at one gene that is often affected by radiation. It's called TGF-beta1. You update, and let's see if that gene was significantly enriched, and it is. In fact, there's a big log change of TGF-beta. And basically, you can see here that the p-value is significant. So not a big surprise. If we go to pair plots, though, this is actually a way to visualize how data reproduce, how well reproducible the data are. So in this case, we're looking at flight rep 1 and flight rep 5. So that's the replicate 1 and 5. So I go back to the PCA plot here. Rep 5 is this point, and rep 1 is this point. So you can see they're pretty far apart on the PCA plot. And sure enough, if I go back to pair plot, you see the correlation is still pretty good, but you see some differences in terms of gene. Now let's play with this, and let's look at the ground control, where we know data were much more clustered. So I'm going to select the rep 1 and the rep 2, for instance. I update the graph, and what you would see is probably a much nicer linear response. Which is the case. Great. All right, let's move on. Volcano plots, it's a way to visualize, compare two groups. So here it's flight versus ground control, and the y-axis is the p-value, log 10 p-value, and the x-axis is the fold change, log 2. And basically, you can actually, over some of these points here, it basically identify for you, if you use the lasso, for instance, you can visualize some of these genes. And those are highly significant. They have a low p-value and they have a large fold change. So that's a way to visualize things if you're interested. You can compare the group also using the dendrogram. It's shown here comparing flight and ground. And you can control the maximum number of genes displayed here and the p-value. You have the classic heat map, and so in this case, flight versus ground. Those are z-score. You can over each of these group, and you can see the z-score and the gene. At the bottom, you have the group, the sample. So you can see that all those are flight. So the flight cluster nicely together, and the ground control cluster nicely together. Again, that's not a surprise. This is supposed to be a strong response. S-plot, that's our way to approach inflammatory response. Pathway response. So you need to pick your favorite pathway. So inflammation is typically change in space. So I'm going to type inflammatory. And the beauty of our system is that it will actually give you options, control vocabulary for the pathway. So I can click inflammatory response, and then now all the genes from the inflammatory pathways are shown here. If they are shown, their p-value is less than 0.05. That's the cutoff we use. I can see more genes by simply going higher in p, but we don't want that. I'm going to actually go lower, 0.03. The fold change is also another factor, so you can actually isolate the genes that you want to look at. So for instance, those genes here are highly significant in terms of p-value, since they met the cutoff, and also highly significant in terms of fold change because they're very high away. So you could look at all kinds of pathways here of interest if you want. The visualization 2.0 will be focusing more on trying to basically look across different data sets instead of just focusing on one data set at a time. Right now, the version 1 can only look at one data set at a time. Okay, one last thing I want to show you before I pass it along to Amanda is, let me go back here and search for GLDS 4. It's one of our original study. I like this one because it's an old microarray data, and then here you can see that illustrates something we're starting to do on all our data sets. We are providing a DOI, which is a digital object identifier. So remember the FAIR principle, findable. The DOI is a critical piece of this findable. If you have a DOI, that means that you can never lose this information anymore online. And so the DOI is actually a prerequisite for being able to cite a data set into your manuscript. So again, I would encourage you, if you use our data, to download the reference, which is available at the bottom of this link here. And that reference download will have the DOI. And so when you cite the data into the paper that you write, then people can really find this data very easily. So the findability is a very important aspect. So one last thing I want to cover is the environmental data tab. So if you click there, we have been focusing primarily right now on radiation dosimetry, but having said that, we are also collecting all kind of telemetry. So let me just emphasize one aspect of it. So basically, if you have a space flight, there are dosimeter on the transportation system that you use that tells us the amount of dose received for an experiment. So we have some special data. They stand for STS, and the special here are shown here. So it's interesting if you look at this table, for instance, you have the GLDS number, which is the ID number of our data set. So you can see that, for instance, STS 135 was one mission. That has actually quite a few data sets on our database. And you can go to each one of them. STS 131 and so on. We have the date of the mission. It also gives you the inclination, so basically where you were located. Importantly, you have the dose that was monitored. So for instance, what's interesting is you have different location, DLOC1, DLOC2, and so on. Those were passive detectors indicated here. And we can see that something that people don't realize is that, for instance, in the same area, you could get a dose of 3.26 milligray and 5.46 milligray, 5.26, 4.66. So you can see those numbers are not the same at all. And the reason is that if you look at here where that's the view of the spatial, it's a panoramic view, and you can see actually the position of these different detectors, DLOC3, 1, 2, and you can see where they were, and pretty much in the same area, but the shielding is quite important in the transportation system, both on the shuttle and also on the ISS, because the cosmic rays are pretty small still. We're still fairly well protected from the magnetic field of the Earth, so you're really not seeing much of a cosmic radiation level yet, and you're seeing what we call low linear energy transfer type of radiation that are not as penetrating and therefore can be affected by the amount of shielding a particle will encounter entering the transportation system. We have done the same for the ISS data. It's a bit more complicated for the ISS because we have really both active and passive dosimeter. If we focus, let's say, on Rodent Research, you'll see that the data are kind of inverted in terms of the way they are displayed. The actual missions are shown as a row this time, and you can actually walk through all these different missions this way. And we have divided the dose into galactic cosmic radiation and a South Atlantic anomaly, which is basically when the transportation system is passing over. Well, the Van Allen belt has a part that comes very close to the Earth, and they call SAA. And so whenever the ISS is going through this zone, you will get a higher dose of radiation. And so that we separate this radiation typically. So that's indicated here. We give the dose and absorb, the dose rate and the absorbed dose. So it's really helpful. You have all this information, play with it. And finally, if you need more information, I would also recommend you go to the Life Science Data Archive at SDA, where also more telemetry are available to you. Thank you.
Hello, I'm going to give you guys a very brief overview of the GeneLab Analysis platform, which you can get to by going to this link here. And any of you can use this platform to analyze space-relevant omics data. So let's say, for example, you have raw RNA sequence data from model organisms that have been flown in spaceflight and also respective ground control. And you want to transform that data so that it goes from something that looks like this, which is FASTQ file containing raw sequence data, into something more meaningful to most scientists, such as a differential gene expression analysis, where you can easily see which genes are up or down regulated in your spaceflight samples relative to your ground control samples. And in this example here, all of the genes that were upregulated in our spaceflight samples are highlighted in red, and all those that were downregulated in our spaceflight samples are highlighted in blue. However, there are several ways for you to get from this raw data to a differential gene expression analysis. And how we decide that is by working with our analysis working groups, which Sylvain had mentioned. So this is the GeneLab RNA-seq processing pipeline. And for those of you unfamiliar with an RNA-seq processing, first we take raw FASTQ files, similar to what I just showed you in the previous slide, and we check the quality of that sequence data. And then we trim that sequence data and filter it based on quality, and then run our FASTQ-C analysis again to assess the quality to ensure only our highest quality data is transferred into the next step, which is an alignment step. And this is where you take that trimmed sequence data and you line it back to a reference genome of the organism from which your samples were derived. And once you line those reads back to that reference genome, your next step is to count the reads to quantify the amount of transcript being expressed in your control and experimental samples. And that'll give you a raw counts for each one of your samples for each gene. You then take those raw counts and you normalize them to control for things such as differences in read depths and transcript length. You next take those normalized counts and you run a differential gene expression analysis to see which genes are up or down regulated in your experimental versus your control groups. And you could do all steps of this pipeline on our analysis platform, which I will go to now. So there's two different ways that you can get to our analysis platform from our home webpage, genelab.nasa.gov. First is by going up here to the Data and Tools panel and clicking on Analyze Data, or you can click right on the interactive button on the home page. This will bring you to a login page, which looks like this. And if you are a NASA employee, you can log in with your NASA credentials by clicking here. Everyone else can log in using a Gmail account. If you don't have a Gmail account, you can click here and create one. And then you can click this button here, input your Gmail account along with your password, and it'll bring you to our analysis platform homepage, which is powered by Galaxy. And I want to encourage those of you who are unfamiliar, have never worked with Galaxy, to click on this Galaxy link here, and it'll show you all the features of the user interface of Galaxy and how to use everything. If you're ever working on the GeneLab analysis platform and you run into any issues that you can't troubleshoot, please shoot us an email at this email address here. So you'll notice that this analysis platform contains 3 distinct panels. The panel here on your left is where you'll find all of your tools or software to analyze omics data. In the center panel, that's your working panel. So here's where you can view files and add options to different tools that you're running. And then over here on the right is your history panel. And this is where you'll import files into, and any analysis that you run, the output data will be displayed in your history. So first things first, before we can do anything, we have to upload the data. There's a couple different ways to do this. One is by clicking this little up arrow, and you could type in a URL, or you can upload files from your computer. We also have data that's already within our analysis platform that you can find by clicking under Shared Data and Data Libraries. We are working on getting access through Galaxy to all of the data in our repository, but in the meantime, we do have a test data set here that you can start working with. And we have each step of the RNA-seq pipeline displayed here. And so if I just want to start with raw data, for instance, I can click on that raw data directory. That raw data link, and it'll show these FASTQ files from this study, which is GLDS 120, along with corresponding FASTQC data. So let's say I want to start with just the FASTQC files. You can either click on files individually here, or if you click on this button, you could click all files, and then you want to bring those into your history by clicking on the Export to History button up here as dataset. And you can either add it to your current history or you can create a new history, which I'll do here, and import. And then you can get back to your main page by clicking on the green pop-up window, or you can click the Analyze Data button. And we're now back to our analysis platform homepage, but you'll notice on the left panel here, our history has been populated with those 2 files that we selected from our data libraries. So now that we have files, we can try testing a tool out. You can either navigate through these different drop-down menus for the tool you want, or if you know the name of it, you can type it in this search menu, which I'll do. So the first step we do with our raw reads is we check the quality. We do that using FastQC. So I'm going to click on that tool here, and you'll notice the center panel will populate with information about the FastQC tool, including FastQC, the name of the tool up top here, as well as drop-down menus to import data from your history and options that are specific to the tool. If you're unsure of what these options mean for any of the tools on the platform, you can just scroll down in the center panel and you'll get more information about each one of those tools. If you're uncomfortable making a decision or customizing it, or you choose to, you can always use the default options that are available within the system. Now if I want to run FastQC, you'll notice that this drop-down menu populates only with files that are in my history. And you can either select just one file, or you can click on this icon here, and you can use your controller command key to select more than one file to run the same analysis on. For now, I'm just going to click here, run it on that first FastQ file, scroll down to the Execute button and click it. And you'll notice that this tool has 2 output files. Gray means that they have been put in the queue. Once your jobs turn yellow, it means the job is running. And once it's complete running, it'll either turn green, meaning that the run went okay, or it'll turn red, indicating that there was an error, in which case you can either try troubleshooting, or you can shoot us an email at the address on the analysis platform homepage. So we're not going to wait for that. I'm going to come here and just show you how to look at our output files. So these are actually the output files from the final step, so from the differential expression analysis. So we do see things such as a differential expression analysis table, which you can see by clicking on any file in your history and clicking on this little eyeball button. So you notice that we have our gene ID, our log to full change, along with our statistics, similar to what I showed you in the PowerPoint presentation. If you want to download this file to your computer, just click this floppy disk icon here, and you'll be able to download it. And just to show you with this differential expression analysis here, in addition to those tables, it also produces images. So you'll see here that we have a couple images, including a PCA plot, which Sylvain had went over earlier, as long as some additional plots and information. And again, if you want to download any of these to your computer, all you have to do is click on this little floppy download disk here. So unfortunately, because I wanted to keep this really short, I don't have much time to get into the details. But I do want you to know that we do have tutorials available if you go to our home page under the Help menu and Tutorials. and scroll down to the bottom, you'll see a link to an RNA-seq analysis video tutorial, which will walk you through how to analyze RNA sequence data from raw FASTQ files all the way through to differential gene expression using our analysis platform. And again, if you ever run into any issues at all or you have comments, please just shoot us an email. We'd love to hear from you. Thank you.